Chromatography is a technique that can be used to separate and purify components of a mixture so they can be analyzed. It has many applications such as developing vaccinations, food testing, drug testing, and for therapeutic uses. We will be using fast protein liquid chromatography in our lab. Different methods of FPLCs such as ion exchange, size exclusion, and affinity can be used to separate and purify proteins of interest within a mixture based on their properties. Proteins are composed of amino acids and can have a net positive, net negative, or neutral charge depending on the side chains. Ion exchange chromatography uses the reversible electrostatic interaction of the stationary phase matrix to separate proteins based on charge. In the case of cation exchange chromatography, positively charged proteins are separated as they bind to the negatively charged beads, while the negatively charged proteins are washed away. Size exclusion chromatography separates molecules based on size and shape by utilizing the porous properties of materials such as beads. Larger proteins are unable to enter the porous beads and elute faster, while smaller proteins can enter the porous beads and therefore elute slower. Affinity chromatography separates molecules based on highly specific and reversible binding interactions to a particular ligand attached to an insoluble matrix. In this diagram, the tagged proteins are separated as they bind specifically to the anti-tag antibody-coated beads, while other molecules are washed away. GST-tagged proteins are fused with glutathione S-transferase. GST tags are used because it allows for purification without denaturation or the use of mild detergents. They are purified from a cell lysate using glutathione affinity chromatography. Purification is based on the strong glutathione GST affinity interaction. GST-tagged proteins specifically bind to the glutathione ligand that is immobilized to a matrix. Because the binding is reversible, the GST protein can be eluted out in a subsequent step. First, an equilibration buffer is added to the glutathione chromatography column and allowed to drip into a liquid waste container. This buffer creates the optimal condition for the GST-tagged proteins to interact with the glutathione ligand. For example, the buffer may contain tris to maintain pH. The second step is to load the sample into the column. The GST-tagged proteins of interest will bind to the insoluble part of the matrix. The third step is to add a wash buffer to remove any contaminants that are bound to the column. The last step is to add an elution buffer containing reduced glutathione. The collected eluate will contain the GST-tagged proteins. Note that the elution should be under mild conditions to preserve protein function and avoid denaturation. For subsequent use and analysis, the GST tag can be removed with a site-specific protease as it may affect analysis. In our lab, we will be performing SDS page analysis to determine the molecular weight of proteins in our sample.